A smidge behind time, we are just going to jump into this. I'll remind our speakers that they have 10 minutes to speak, 2 minutes for questions. When you see the yellow flashing, that means it's time for questions. When it turns red, we will kick you off the stage. With that, I am happy to introduce our first speaker. This is Richard Gilliland, and they'll be speaking about a structural proteomic survey of Thermus thermophilus under deep ocean pressure.
Thanks a lot. Can you hear me okay? Great. All right. I just want to quickly give a pitch for the Cornell hydrogen synchrotron source. We have an X-bio beamline, actually 2 beamlines, that specialize in high pressure and extreme conditions. So if you're interested in X-ray scattering, crystallography, under extreme conditions, come see us. We have a great team, and it's easy to write a 1-page proposal. You don't have to pay anything, so give us a try if you're interested. Just so everyone's on the same page, I know not everybody thinks in terms of megapascals, but just as a reminder, since we're talking about pressure, that 1 megapascal is 10 bar. Can you see my pointer okay? Yeah, great. Is 10 bar or 10 atmospheres, okay? And the wreckage of the Titanic is at 40 megapascals. That's the abyssal plain, as you all well know, that's a large percentage of the Earth's surface. And the deepest points in the ocean are about 100 megapascal, the Challenger Deep. So when I talk about terrestrially relevant pressures, I'm talking about 0 to 100 megapascals. All right. There are places, possibly on the Earth, where there can be much higher pressures and living things. In the subduction zones, it has been estimated that pressures may get up to 340 megapascals and still be compatible with life, okay? But of course, we're all here because of ice worlds, right? Now, Enceladus, these are all estimates of pressure with big question marks, of course, is a small planet, 5 megapascals, so not a terrifically high pressure. Europa is much more interesting from the pressure biological perspective. And then, of course, Titan, there's a very big range and potentially huge pressures. Okay. So the one thing to remember about the effect of pressure on biomolecules is you think about free energy as determining minimum free energy, determining what the conformation of a biomolecule should be, and the change of that free energy with pressure is equal to volume. And so the bottom line is that pressure favors smaller total molar volumes. That includes solvent. And the one thing that I would like you to take away from this whole symposium is anything a biomolecule does changes its volume. Let me just get further into that, right? So the volume of a biomolecule you can think of as a collection of spheres, hard spheres, and then there's an excluded area that solvent cannot reach, right? And so if this molecule were to be pressurized, of course it could shrink to reduce its volume, but actually the easier thing may be to unfold so that you expose the cavities to solvent. And that's kind of paradoxical, right? That something should transform into what looks like a bigger shape under pressure. But that's well established that phenomenon happens. And in fact, the big picture that's kind of emerging is that molecular complexes, if you put them under pressure, ocean pressures, they tend to dissociate, and then they start to unfold as you really put pressure on them. I do want to give a really quick pitch to call out to Jacob Winnigoff, who's here in the audience. Jacob and Itai have a science paper that will shortly be coming out, which talks all about the importance of lipids, which I can't really get into, in ctenophores. And it's a really beautiful piece of work. If you take a ctenophore from the deep ocean, you bring it up, it kind of melts, and there's an incredible story to do with lipids and phase transitions. So please be on the lookout, track him down. He's got a poster tomorrow in the poster session. So I'm going to talk about proteins. This is a scattering profile of a typical standard protein I pulled off the shelf, and I did many of these when we started the high pressure work and was kind of horrified to discover they don't do very much, at least on the large scale when you pressurize them. So what we really need is something like when people are searching for historic wrecks and things on and ocean hydrothermal vents and whatnot, some sort of a way of mapping the entire surface, the whole vast sea of proteins, to find where the treasures are hiding. And so I was at the RCN, this is a research coordination network for extreme biology supported by the NSF, and I met Haley Moran, who's a graduate student of Stephen Fried, at Hopkins. And as I started to learn about her technique, I realized, whoa, this is exactly what we need. This is the map that will lead us to the treasures. And so here's the basic idea. If you take a protein that has some secondary structure, you pressurize it, and there's some sort of a conformational change that exposes a secondary structure element to the solvent, and then you add proteinase K, which is sort of an indiscriminate cutter of peptide bonds. It will go on the surface and it will cut what sticks out. So you'll get a cut point, and then you can then sequence that with proteomic techniques and figure out what part of the structure changed. All right, so this is the basic. Here's the team. Whoops, sorry. Here's the team. Edgar is the bioinformatics person. Shreya actually runs the machine remotely since I'm up in Cornell and they're down in Hopkins, okay? And this is the part, we don't really have time to get into the instrumentation that it took to do that, but I developed a system that allowed them to do that, okay? And when we ran I was expecting a few percentage change in the proteome. And when we saw the results, I had to go back to them and say, come on, you got to check your statistics. This cannot be right. At 100 megapascal, we found that 39% of the proteome of Thermus thermophilus, right? So this is an organism that lives in hot springs, but it doesn't like pressure, so it's pressure sensitive. We found that 39% of that proteome is sensitive to pressure at least one site, okay? And I can't get into the details. We do sort of a broad survey of mostly cytosolic proteins, but we also get ribosomal associated proteins, which I can't really talk about today. But the most exciting thing, I think, is if you look at, sorry, I must have timing on. If you look at the different kinds of cofactors associated with the proteins, you'll find that highly charged metal ions tend to destabilize proteins with respect to pressure. Also, what we find is that if you move away from a neutral charge state to a protein that's charged at a particular pH, it also will become more unstable. So there's definitely a charge something going on here. And also we measured, this is something that Stephen had to convince me was a sensible thing to do, but it turns out it's really a remarkable result. If you measure something called the packing density, and we've measured it by a couple of different means, what you will find actually is that as the protein becomes more packed, denser, it tends to be more pressure sensitive, and that is completely contrary to what we expected on the basis of the experience that we had so far with individual proteins. And I think there are a couple of hypotheses that we can put forward, and that is that maybe loose packing does allow you to reshuffle your atoms a little bit to avoid pressure sensitivity, but tight packing is probably necessary for proteins to actually perform their function. They need to move in a predefined way, allostery and so forth, and so maybe that has something to do with what we're seeing here. Okay, so I'm just going to do a quick survey of some of the actual things that we saw. So I picked out of the pile enolase because I know, at least in the yeast enolase, what that does under pressure. And these magenta sites, if I can get them to hold still, sorry about that, are the places where residues have become more pressure accessible. And you'll see that here's the phosphoglycerate and a couple of magnesiums. I thought that's pretty interesting. It picks out the active site, the active pocket. And this happened again and again and again. Here's another one, here's another one, here's another one, right? It seems to be picking out active sites involved with charged species. Here's another one. This is actually an antibiotic that is not a natural ligand, and you'll notice there's nothing around it. It's around the natural ligand that these pressure-sensitive changes appear. So here's one that seemed to buck the trend, right? There's a citrate and a GDP, and there was nothing highlighted. And then I realized, well, what does this thing really do? These proteins form ropes around cells that sort of tighten during cell division. So we really need to build a model, and fortunately those exist. And you start to see some really interesting patterns going on. And sure enough, when the active site, if you look at the whole model, there's that potassium, there's that charged site, and the pressure-sensitive areas. Okay. Here's another one which is fundamental to the citric acid cycle. It appears to be breaking apart at the helices. And if you look at the ligands and compare known structures, what you can see is definitely there's something going on with that magnesium. It's actually calcium in this state, but it can be a manganese or a magnesium depending on the environment. And so clearly there's something to do with pressure ejecting or modifying these charged states. And that is a natural thing to believe because charges, as we know, have something called the electrorestriction effect that compresses water. I think I have to wind up here. And it's a good point to wind up. This is a veritable treasure trove of things that we're discovering, curiosities of all different kinds as we probe. And this Thermus thermophilus is a small proteome. It's only a couple thousand proteins. And so as we get into bigger and bigger proteins, we really have an enormous cache of interesting structural biology on our hands that is clearly involving charge and electrorestriction. So with that, thank you very much, and we'll take questions.
Thanks, Richard. I think we have time for